For most of history, a type 1 diabetes diagnosis was fatal. It occurs when the body stops producing insulin, the hormone required to process glucose. By the 1920s, doctors found a brute force workaround, scavenging insulin directly from the pancreases of slaughtered pigs and cows. This extraction process was highly inefficient. It took nearly 2 tons of pig parts to produce just 8 ounces of usable insulin. Medical drawbacks were common. Because animal insulin differs from the human version by a few amino acids, patients frequently developed immune reactions that decreased the drug's effectiveness over time. Facing an unsustainable supply chain, researchers in the 1970s asked, could we bypass animals entirely and teach a single-celled organism like E. coli to manufacture a human protein? Solving the insulin shortage required medicine to transition from an extraction industry to an engineering industry. The process starts by isolating the exact sequence of human DNA that codes for insulin production. Next, scientists need a biological delivery vehicle. They use a plasmid, a small circular piece of DNA naturally found inside bacteria. Using chemical tools called restriction enzymes, researchers cut matching sections out of both the human DNA and the bacterial plasmid. The cut ends connect through natural base pairing, and an enzyme called DNA ligase permanently seals them together into a continuous ring. This newly engineered ring is then physically inserted back inside a living E. coli cell. This step is called transformation. That single modified bacterium now holds the complete genetic blueprint to manufacture a protein its species has never produced before. This diagram illustrates the specific engineered plasmids used by researchers. Notice that they do not allow the bacteria to create fully formed active insulin. Instead, they isolate the insulin A chain and the insulin B chain into separate plasmids. Because human insulin is composed of these 2 distinct chains, they are intentionally grown in entirely separate bacterial batches. This is a strict precaution to ensure no active biohazards are accidentally produced during cultivation. Once the cells produce the raw components, the individual chains are extracted and purified. Only then are they chemically assembled with a disulfide bond to form active insulin. How do you turn one engineered bacterium into a global medical supply? The answer is fermentation. Placing the bacteria in massive, nutrient-rich bioreactors triggers exponential multiplication, creating trillions of microscopic factories. This decoupled production logic allowed for massive industrial scaling, while eliminating the risks of both active biohazards and animal impurities. In 1982, the FDA approved Humulin. Patients now had access to a highly scalable supply of a drug that was chemically identical to human insulin. The production cycle is precise and repeatable. Isolate the gene, cut and join the plasmid, transform the bacteria, ferment the cells, and purify the protein. This cut, paste, and culture mechanism solved the insulin shortage, but it also functioned as a highly reliable platform technology. Researchers applied this exact same bacterial mechanism to produce safe human growth hormone, blood clotting factors, and eventually targeted antibodies for cancer therapies. By teaching a simple bacterium how to read and execute human genetic code, scientists launched the entire modern biotechnology industry.